Assalamu alaikum. This is Uzma Batul from the Department of Chemistry. I, along with my group members Zoya Safdar and Meera Mudassir, are here to discuss with you the applications of titrations in the pharmaceuticals. The use of titrations for the analysis of the organic compounds continues to play an important role in the pharmaceutical, biological, agricultural, and the environmental laboratories. The pharmacists use these titrations to get the proper mix when they compound a medicine and to know about the exact composition of the different medicines. If we come towards the titrimetry for the analysis of pharmaceuticals, the first application is the determination of the ferrous gluconate in iron tablets by the redox titration. In this, the titrant used is the cerium sulfate, this yellow compound, and the ferrous gluconate. These are the ferrous gluconate tablets, and here the structure of ferrous gluconate is shown, and the indicator used in this process was the ferroin indicator. Gluconate is a type of iron that is used to treat the iron deficiency, and it usually contains about 12% elemental iron. In this process, the titrant is the ceric sulfate and the titrant is the ferrous gluconate. The indicator used is the ferroin indicator, and the color changes at the end point as red to pale blue. Coming towards another application, that is the determination of the tocopherol by redox titration. Tocopherol acetate or the vitamin E acetate is also known for its the antioxidant properties. Its structure is shown in this figure. Titrations. The titrant used is the ceric ammonium sulfate. Titrant is the sample containing the tocopherol, and the indicator used is the diphenylamine. The diagrams of all of the three are shown here. If we come towards the procedure of the determination of tocopherol, here the 50 ml of the saponified sample is mixed with the 50 ml of alcoholic sulfuric acid and about 20 ml of water. Then to this solution, the 2 drops of indicator diphenylamine solution are added and is then titrated with about 0.01 N ceric sulfate solution. Determination of menadione by the redox titration also has the application in pharmaceuticals. The menadione Vitacate tablets are used to treat the hemorrhages, you can see in this picture, and it is also called as vitamin K3. The structure is also shown, basically contains the 2 benzene rings with the branchings of the oxygen and also a methyl groupase attached. The titrants for the menadione determination are the titanious chloride and the ceric sulfate solution. The titrant is the sample that contains the menadione, and the indicator used is the O-phenanthroline. The diagrams of the indicators and the titrant and the titrants are all shown in the pictures, and here you can also see the structure of the indicator. Determination of menadione is also redox titration, that is, it involves oxidation reduction reaction. Firstly, we titrate it with a strong reducing agent, that is titanious chloride, and it involves the reduction with zinc and HCl. Then the reduced form is titrated with the ceric sulfate using O-phenanthroline, that is the indicator used to assay it. The determination of sodium 2,6-dichlorophenols also occurred by the redox titration. It is basically an indicator dye. Here you can see the structure of the sodium dichlorophenol, salt of sodium dichlorophenol, and this is the indicator. It is used in the determination of the ascorbic acid in the pharmaceutical products as well as food, and in other cases when the titration of vitamin C is not possible with the iodine or any other method. The ascorbic acid, when it reacts with the indophenol that is initially red in color, it is converted to the oxidized vitamin C along with the reduced indophenol, which is now the colorless complex. It can be assayed by titrating the iodine liberating from the acidic solution of dye upon addition of the potassium iodide. In other words, the substance is oxidized. A substance oxidizes the HI in an acidic solution to iodine and is then titrated with the standard solution of the sodium thiosulfate. The determination of Valency chloride hydrochloride in the pharmaceutical by the redox titration. This drug is basically used in the treatment of eye infection in the AIDS people. The structure of drug is as follows. It contains rings of nitrogen and carbon. Also the oxygen is attached. along with the HCl hydrochloric acid and the moles of water. This you can see the You can also see the Wilgensi chlorine tablets along with the eye infection in the AIDS patient. The first method of determination of this, the iron 3 is reduced to iron 2 by the Wilgensi chlorine hydrochloride, and it's led to the formation of the iron 3 ferricyanide complex, which is basically the Prussian blue in color. And in the second method, which is actually related to the redox titrations, the permanganate is reduced by the VLGH and it is converted to bluish green manganate in the alkaline medium. You can see the color changes as well. The procedure for this method is also a simple one, that is different aliquots, that is 0.2, 0.51 and respectively others, of the 100 microgram per milliliter of VLGH solution were placed in a series of 10 ml calibrated flask, and the volume was brought to about 4 ml with water. Then 1 millimeter of 0.1% weight by volume permanganate solution was added to each of the flasks, followed by the addition of 1 milliliter of the 5 molar sodium hydroxide. Then each flask was made up to the mark and mixed well. The flasks were kept aside for about 15 minutes and the absorbance of each flask was then measured. The absorbance was measured to be maximum at about 610 nanometers. Now this diagram helps us further to understand this reaction, that is Wilgensi chlorine hydrochloride. It reacts with the hydrochloric acid and the ferric. It is oxidized and the ferric is reduced to ferrous, that is Fe plus 3 is converted to Fe plus 2. Now this oxidation product and the ferrous are further reacted with the hydrochloric acid along with the potassium ferric cyanide, and it results in the formation of the iron ferricyanide complex, which is Prussian blue in color, measured at about 730 nanometers. In another method, alternative method, the Wilgensi chlorine hydrochloride is reacted with the sodium hydroxide along with KMnO4, and it results in the formation of the oxidation product of VLG along with the manganate. Here this is the bluish green in color, that is measured at 610 nanometer. Both of the products are different and are measured at the different wavelengths.
Assalamualaikum, I am Neeraj Mudassar and I will discuss about the determining isoniazid by redox titration. Isoniazid is basically anti-tubercellular drug, and in pharmaceutical industry we assay it by bromometry. This picture shows the structure of isoniazid. Here is the method of determination of isoniazid. So it is determined by the addition of potassium bromide and the direct titration of potassium bromide in the presence of HCl solution. At the end of titration, bromine is released, which further reacts with isoniazid. This is the equation showing the reaction. KBrO3, which means potassium bromide, is reacted with bromine ions in the presence of HCl solution, which produces bromine gas, potassium chloride and water. The indicator used here is methyl red, and at the end of titration we determine the end point as it decolorizes the solution. Next is the determination of polyether derivatives of anthraquinone in medicine, which is determined through complexometric titration. Here comes the brief introduction of anthraquinone. So basically, anthraquinone is a polycyclic aromatic hydrocarbon which is derived from anthracene. Anthraquinones with an ionophoric unit may be considered as precursors of a molecular recognition systems. The pKa values of the investigative compounds carries two information. The first one is that it measures the ability of molecule to complexate metal ions, and the second one is that it determines the proteolytic from taking part in those processes. This graph shows the spectrophotometric observations which are observed during the complexing reactions. The changes in the intensity and position of the absorption band after the addition of metal ion to the ligand solution allow to specify which ions are the most effective and interacting exclusively with the ligand. From this we determine that the group of metals such as copper, cadmium, iron, nickel, calcium, mercury, zinc and aluminium affects the absorption spectrum of the tested anthraquinone derivatives, and the biggest changes in the absorption spectrum of all the analyzed ligands were induced by copper, mercury and aluminium ions. The next one is the determination of aluminium salts in drugs. coagulants, and fertilizers by complexometric titrations. Here is the procedure for determination of aluminium salts. So, in the first step, the sample of aluminium ions are heated with the excess of EDTA, ethylene diamine tetraacetic acid, at the pH of 7 to 8. Then, in the second step, the solution is adjusted at pH 10 using the ammonium buffer. In the third step, the residual EDTA is titrated against the standard solution of zinc ions using iochrome black T indicator. Last, the color change from blue to wine red. This below picture shows the color changes which change from blue to wine red, and the above structure formula shows the structure of EDTA. The next is the determination of methylene diphosphonic acid, which are determined through the complexometric titrations. The complexometric titration of thorium with methylene diphosphonic acid is carried out. Here, methylene diphosphonic is used as the trichromatic agent for thorium. In the presence of trans 1,2-diaminocyclohexane, a soluble binuclear ternary complex is formed. The slightly acidic medium is used for titration, which is further buffered with ethylene diamine EDA. And the indicator used here is methyl mole blue. For the determination of MDP in labeling kits, we use two 5 ml aliquots of thorium nitrate solution, which are respectively titrated with 2 mm MDP, and the MDP solution is further prepared from the labeling kit. After this, a soluble binuclear ternary complex is formed, and methylene diphosphonic acid also forms a similar complex. A sharp endpoint is obtained with a color change from blue to colorless or smoky gray. Here are some applications of MDP. Methylene diphosphonic acid is a superior agent for skeletal imaging. Labeling kits contains 5 mg of MDP, 0.5 mg of stannous chloride, 9 mg of sodium chloride, and 1 mg of 2,5-dihydroxybenzoic acid. That's all from my side. Thank you so much.
Assalamualaikum, here's Safdar. I'm further going to discuss about the determination of different ingredients in pharmaceutical products. One is the determination of active ingredients in tablets. Titration is used to determine the content of active ingredients in pharmaceutical products like acetylsalicylic acid in aspirin and vitamin C in multivitamin tablets. Determination of acetylsalicylic acid, which is an active ingredient in aspirin tablets, which is used as an analgesic, reduces pain, and is anti-inflammatory. This is the structural formula of acetylsalicylic acid, which is white in its pure form. This is the chemical equation showing the formation of acetylsalicylic acid from salicylic acid and acetic anhydride in the presence of sulfuric acid. These are the aspirin tablets. Analysis of aspirin tablets are using titrant and sodium hydroxide, which is white in its pure form. This is the structural formula of sodium hydroxide. The indicator used for this analysis is phenolphthalein. This is the structural formula of phenolphthalein, and this is the balanced model of phenolphthalein. Equation for this analysis include that number of moles of sodium hydroxide are equal to the number of moles of acetylsalicylic acid. Next, action of phenolphthalein. Phenolphthalein indicators show different color at different pH range. At a pH of 0 to 3, it shows orange color. When the pH is from 4 to 7, it is colorless. And when the pH ranges from 8 to 10, it becomes pink. And when the pH is highly basic and ranges from 11 to 14, it becomes colorless again. Normal percentage in aspirin tablets. 37 gram of aspirin contain 8.92% of acetylsalicylic acid. If only about 9% of aspirin is an active ingredient in its use, then the rest of it is made up of other substances, probably to give shape and appearance. Analysis of pharmaceutical compounds. Many pharmaceutical compounds are weak acids or bases that can be analyzed by an aqueous or non-aqueous acid-base titrations. Examples include the analysis of phenobarbital, caffeine, and sulfonylamide. This is the structural formula of sulfonylamide, which is white in its pure form. This is the phenobarbital tablets, and this is the caffeine and with its structural formula. The procedure include titrating samples using sodium hydroxide and phenolphthalein and methyl orange acting as an indicator. This is the apparatus setting for the titration. Next is the purity analysis of pharmaceutical products. Acid-base titrations are also used for checking and determining the purity of many medicines and drugs. Examples here include the purity control of ephedrine hydrochloride. This is the structural formula of ephedrine hydrochloride, which is white in its pure form. Uses of ephedrine hydrochloride. This drug is used in the synthesis of many cough syrups in the treatment of bronchial asthma. The procedure for the analysis of ephedrine hydrochloride include that sample is dissolved in inorganic solvents like anhydrous acetic acid and mercuric acetate, and then the solution is then titrated using perchloric acid as a titrant. This is the structural formula of perchloric acid, and this is the perchloric acid in its liquid form. These are the chemical reactions involved in the analysis of ephedrine hydrochloride using acid-base titration. Determination of barbiturates. Barbiturates are central nervous system depressants. All barbiturates affect the gamma aminobutyric acid, a neurotransmitter. This is the structural formula of gamma aminobutyric acid, which is acting as a neurotransmitter as shown in the following image. The example of barbiturates include butabarbital and secobarbital. The procedure for the analysis include that sample is dissolved in methanol and water mixture. Indicator is added in the sample solution, usually phenolphthalein. The solution is then titrated using strong base like sodium hydroxide. The next one is the determination of nicotinic acid. It is also known as niacin, which is vitamin B3, which occurs naturally in food. This is the structural formula of nicotinic acid. Used as a medicine, it improves levels of blood fats such as cholesterol. It works by reducing the amount of cholesterol and triglyceride made by liver. This is the image showing that nicotinic acid is also used as a medicine. The procedure here includes that solution containing niacin is titrated using sodium hydroxide as a titrant and phenolphthalein as an indicator. The equation here used is the same, that number of moles of sodium hydroxide are equal to the number of moles of niacin. A question arises here that how titration is used in medicine. Titrations are used in medicine to limit side effects, taking time to see how our body will react to a drug. Medication started at low dose every couple of weeks. Dose is raised means that the concentration of active ingredient is raised in the dose until maximum effective dose, which is known as target dose, is reached and no side effects are observed. Drugs which are usually titrated include insulins, antidepressants, anticonvulsants, and sedatives. Titrating of the medication instead of stopping abruptly is recommended in many situations. pH stating. Stating maintain a defined pH value during a defined time by controlled addition of either acids or bases. Applications here include the determination of enzyme activity, quality control of many medicines, checking the kinetics of chemical reactions, and determining the buffer capacity of antacids. First one is the determination of enzyme activity. Enzyme reactions that uses up or form H positive are monitored using tensiometric electrodes. The H ions formed are used up or neutralized by the controlled addition of either alkali or acid respectively, and pH is controlled constant. The rate of titrant addition is proportional to the reaction rate of enzyme under investigation. Determination of chemical activity of lipase enzyme is an example of it. The last one is the determination of buffer capacity of antacids. Antacids are substances which are used as therapeutic agents for neutralizing excess acidity in stomach and intestine caused by gastritis and intestinal disorders. Examples here include the magnesium hydroxide and aluminum hydroxide. This is an image showing the excess acidity in stomach. These are the antacids tablets. Antacid must keep the pH value constant during its residence time in stomach. Neutralization capacity, reaction rate, and buffer capacity are determined by pH setting process. Thank you.